Okay, so this is the section that's going to start leading into the physiology of the nervous system. And so in order to start understanding the physiology, you have to understand the plasma membranes of neurons. Um, and so we have to start talking about the pumps and the channels that are in the membrane. And so there are special membrane proteins, and they work just to maintain a concentration gradient, and they are going to move substances on one side or the other to make sure that they stay in the proper locations. In order to do this, you have to spend cellular energy. And so just like with muscles, you have to push sodium and potassium into the proper locations. With your nerves, you're going to have to push sodium and potassium into the proper locations, and even calcium as well. And this is going to all work to help maintain the proper function of a neuron. And so we talked about pumps. Those are going to move things against their concentration gradient and use cellular energy. And this is different from channels. And channels are going to be proteins that allow substances to move down their concentration gradients. So once you establish a concentration gradient with a pump, you can then use that energy as a channel later to help get a desired reaction. And so there are a few different types of channels. You have leak channels, and these are going to always be open, and this allows for continuous diffusion, which might seem kind of counterproductive, but we'll see instances where it's necessary. There are chemically gated channels, which are typically going to be closed except for whenever they're bound by a neurotransmitter. And so this is similar to the acetylcholine channels that we saw at the neuromuscular junction. And then there are voltage gated channels, which are going to be closed except for whenever the membrane charge changes. And this would be like those sodium potassium channels that help propagate the signal down the muscle or down the sarcolemma. And so this is going to be channels present in neurons. And so here are some different types of pumps and channels. Or here are some channels that we'll see. These are sodium channels. And so sodium channels have two gates and can be in three states. And so the three states, the first state is the resting state. And in rest, they're going to be closed. And so notice here, here's one gate right here, the inactivation gate. And at this point in time, it's open. And so I'll show you how you can tell it's open. Y'all can see this little space here in between. And so this little space in between is going to be where something can go through. However, in the resting state, the activation gate is closed. And so that's like this little flap that sits here on the outside of the channel, which is actually on the inside of the cell, and it prevents substances from being able to pass through. And so you can see here, the second state is the activation state. And so during the activation state, the inactivation gate still is open, and the activation gate is also open. And so now this means that substances can be able to go through freely. There's nothing preventing them. And they're going to go down their concentration gradients. And so if this is a specific sodium channel gate, or a sodium gate, it's not going to allow any other ions to go back and forth, just sodium. So if sodium is typically on the outside of the cell, whenever this gate is open, that sodium is going to be able to go into the cell. And so what this is going to do to the charge is very important. And so typically there's more sodium outside. So that means there's more positively charged substances on the outside of the cell. But whenever the gates are open, these positively charged substances will then have the ability to go into the cell, and this is going to change the charge of the inside of the cell from negative to positive because of all the positive charges coming in. And then the third state that these channels can be in is an inactivation state. And so after it has reached a certain point in time, or a certain charge, or whatever its stimulus is, the inactivation gate is going to close. And it's the closure of the inactivation gate that provides or forces this channel to reset itself. And so a channel can't become active again until it goes back into its resting state. And so this is going to force a period of time where nothing can pass. Now that period of time is not very long, but it's necessary to have that inactivation state before you can have another resting state, an activation state following that. So here's some text that just describes what is happening here. So there are different types of channels as well. And so modality gated channels are going to be normally closed, but they're going to respond to some kind of stimulus that's not chemical or voltage. Typically it responds to things like pressure or touch. And so this is going to be like, for example, on your skin. Whenever you touch your skin, you're able to feel that. And that's because there are modality gated channels in there that are going to be stimulated by that touch, and that's going to send that information or start that synapse. It's going to send that information up to your central nervous system. So now we're going to talk about where these are distributed or where they're located. And so there are some types of channels and pumps that are located across the entire plasma membrane of the neuron, including the soma, the axon, etc. So there are going to be leak channels, and these are, for example, like potassium leak channels that just constantly allow potassium to leave the cell. Or, for example, it could also be sodium leak channels that constantly allow some sodium to enter the cell. There are pumps, and these pumps are sodium-potassium pumps. And so what happens with these sodium-potassium pumps is they are going to take and pump, make sure that it grabs 3 sodiums and pushes it out of the cell, and it grabs 2 potassiums and pushes it into the cell. And so this exchange 3 out for 2 in, so it's going to be 3 positives going out of the cell and 2 positives coming into the cell, is going to be what helps contribute to the negative resting membrane potential of that cell. And so these leak channels and these potassium pumps are going to constantly be working to help maintain the cell's resting membrane potential. So there are also some areas of the cell that have different types of pumps, and so we need to identify these different regions of the cell. So there's a receptive segment of the cell, and at the receptive segment of the cell, which is the dendrite and the cell body, there are chemically gated channels, and these are gated for substances like chlorine, or they could be gated for neurotransmitters or something along those lines. But it's going to be something needs to stimulate them chemically to enable those channels to open. At the initial segment, which is going to be at the axon hillock, and so remember there's the cell body, and so I'm drawing dendrites, or trying to. Here's the cell body, and then this is going to lead down to that axon. This little bulgy region right here is the axon hillock, and the axon hillock is also called the initial segment. And in the initial segment there are going to be a lot of voltage-gated sodium channels and voltage-gated potassium channels, and these are going to be necessary to help depolarize and repolarize the cell, particularly the axon, since the axon is sending or going to send the impulse down the axon. So there's the conductive segment, which is going to go down the axon and all of the branches that come off of that one axon. And again, this is going to have voltage-gated sodium and potassium channels, and they're going to function similarly to how the muscle did whenever you were propagating that signal, like that domino effect, where the sodium channels will all open and make the cell become more positive, and then the potassium channels will all open following that to repolarize the cell and make it more negative again. And at the very end, at the synaptic knob, where you transmit this information to the postsynaptic structure, there's going to be a bunch of voltage-gated calcium channels. And this is so that way that charge that's being sent down the axon will now stimulate these channels, and that will allow calcium to enter into the cell, and then allowing that to push those vesicles towards the synaptic cleft. So here is an image that summarizes everything. And so what it shows here is the entire neuron is going to have the sodium-potassium pumps, sodium and potassium leak channels. On the receptive segment, which are all the dendrites and the cell body, there will be chemically gated channels, chemically gated cation channels for positively charged ions, chemically gated potassium channels, and chemically gated chlorine channels. And then there are going to be, in the initial segment, which is just this region right here at the axon hillock, there will be voltage-gated sodium and potassium channels. And then the conductive segment will have voltage-gated sodium and potassium channels as well. And then whenever we get down here to the transmissive segment, there are going to be voltage-gated calcium channels and calcium pumps, which are going to constantly be pumping calcium out of the synaptic bulb.